Hello students, this is Dr. Fazan Mirza. In this video, I'm discussing some P4 questions from selection and evolution. So let's get on with this. The first question that I'm taking is from October-November 2021, variant 42. So this fish here, they are telling this is in that this basically 20 billion years ago, an ocean covered the area where the country Panama is now located. There was a gap between the continents of North and South America through which the waters of Atlantic and Pacific Oceans flowed freely. The pork fish, which is shown here, the name is provided to you, lived in this area between North and South America. This is what's happening here. About 3 million years ago, you can see volcanic activity and sedimentation formed a narrow strip of land with Panama, joining North and South America. So you can see there's North America, there's South America. 20 million years ago, whereas this is the condition now. So now you can see there's a land Panama, which is connecting the two continents together. So whereas previously this gap was there. So Pacific Ocean and Atlantic Ocean, the fish would have flown basically swam freely, not flown. So yeah, it would have moved freely from Pacific Ocean to Atlantic Ocean. Yes, but now because of this land being there, the fish in Atlantic cannot cross over to Pacific, and the fish in Pacific cannot go over to Atlantic. So this is what actually we know. 20 million years ago, poor fish, Atlantic and Pacific, were able to breed successfully to produce fertile offspring. Explained by Atlantic porkfish and Pacific porkfish are now not able to breed successfully to produce fertile offspring. So we need to mention allopatric speciation. We mentioned there's no gene flow occurring. Reproductive isolation has they both have been reproductively isolated. Adaptation is basically will be based on the selection pressure which are prevalent over the years in both the oceans, the Pacific one and the Atlantic. And new mutation would have occurred by chance, and better D would have been selected and passed on to the next generation. So this is just one part of question, but a lengthy question. Sorry, my voice just break. My voice just broke. So yeah, so that's basically just for the 5 mark question, but a descriptive one. So let's move on to the next question. This is Major 22, variant 43. So this is Puma, shown to you in North and South America. Figure 5.1 shows Puma. Distribution of Puma is shown in figure 5.2. So this is 5.2 figure, and we can see that all these area which are highlighted are the one which where Puma is distributed. And there's this region in Florida, which is next to Texas, close to Texas. There's this population of Puma there as well. There's a region B and there's a region A, which are towards the end of the belt where Puma is present. So, and they are pretty distant from Florida and Texas, which you can see from the map here. So let's move on. Members of different subspecies belong to the same species but have some morphological differences are found in different geographical locations. Morphological we know basically means physical and structural changes. So in the past, Puma has been divided into 32 subspecies based on some differences in the morphological features. These subspecies of Puma varied in body size, coat color, behavior, to adapt to each population, its environment. From region A to B, we can see it's a long area, a very long stretch of land. Explain how the different subspecies of Puma evolved. So we know that different selection pressure in different environments would be prevalent from region A to region B. Different prey available, different camouflage or predation required for these for protection of this animal by others or while hunting for its prey. Mutations occurred over time, new alleles that were expressed, they were more suited to the environment. Different populations of Puma subspecies do not interbreed because morphologically they are different and their behaviors might be different. So yeah, they might not crossbreed. In 2016, genetic analysis concluded that there are only two genetically distinct subspecies of Puma in North and Central America, North Central America, and one in South America. Outline the practical techniques. So they are asking you to outline multiple techniques here that could be used to conduct a genetic analysis of Puma species. So what will you do? You will take blood samples from different species and DNA will be extracted from WBCs. The DNA will be amplified using PCR. The DNA fragments the DNA can be fragmented using restriction enzymes. The DNA fragments can then be compared on gel electrophoresis. Microarray gene probes can be used for hybridization of the single-stranded DNA, and the database available on bioinformatics can be used to compare the DNA and the protein sequences to basically see how many different types of genetically distinct Puma are there. In figure 5.2 shows different location of puma evolved in Florida. 1990, the size of this population was very small, very few, smaller than 30 individuals only. So that's a small population. Three phenotypic features that vary in pumas are in the shape of tail, pattern of hair on the back, and the position of the testes in male pumas. Variant forms of these phenotypic features that are normally rare occur at high frequency in Florida, which is a small population. We can expect that this is kind of a population which is semi-isolated, pretty far from the wild population. And these rare variants are common there. The bent tail, abnormal pattern of hair on back, testes remain in abdomen, undescended testes. So that basically would make the puma infertile because if the testes are not descended, the sperms won't be made there. So predict with the reasons whether these phenotypic features show a continuous or discontinuous variation. So yeah, there's a discontinuous variation because these are discrete categories. There's no range of phenotype and no intermediates, either this or that. It's so that's pretty straightforward. Explain how the small size of Florida population resulted in high frequency of these normally rare variants. So why are these rare variants common there? Because it's a small isolated population. So probably a result of bottleneck, genetic drift might have occurred. They do not interbreed with other populations. They are reproductively isolated. No gene flow. It reduces the genetic diversity because we know when the reproductively isolated population form or there's a bottleneck, genetic drift occur and the reduced genetic diversity is resultant in recessive traits accumulating, homozygosity prevails. This reduces the gene pool and a less fit population is produced. In 1995, eight puma female from Texas were introduced in Florida to increase the breeding success of the future size of puma population in Florida. In the next 20 years, the population grew substantially. Suggests why they introduced females were taken from Texas and not from A and B. So we know pumas in Texas and Florida are likely to be adapted to similar environment because they are pretty close, they are nearby. Whereas point A and B on the map, they were pretty distant from Florida. The selection pressure would be not be very similar in those regions, but in Florida and Texas, the selection pressure would be very similar. So morphology would be similar, and they would be likely to successfully breed and mate. And that's actually what happened because they say that this thing was a success. May June 2023, this is variant 41. A genus of grass plant is shown, a food crop. Farmers flood the field, encourage faster growth. You are expected to recall Arenkayama. It allows oxygen for aerobic respiration, submergers. They have direct contact with air. State one other way in roots are adapted to grow in submerged water. Alcohol dehydrogenase enzyme is there, so they can tolerate high alcohol concentration. Another adaptation in some variety of rice, selective breeding would produce varieties of rice fast growing. How selective breeding is carried out? So for selective breeding, we need fast growing stems of rice. So what will we do? Varieties which are already growing fast, they will be selected and they will be bred with the rice crop that we have because we want the fast growing ones to pass on the genes to the offspring that that we will be producing with them by crossing it with the rice crop that we have. The offsprings of these are observed, and those which are growing the fastest are the ones which are selected for further inbreeding. And basically, we carry this out for various generations. Outcrossing is carried out for avoiding the homozygosity because recessive traits may accumulate. So that's how we carry out selective breeding. They're asking you how auxin works, so you can recall the effect of auxin, how it works by binding on the cell surface membrane, allowing H plus ion to be actively pumped out. This breaks the hydrogen bonds between cells, microfibril, potassium enters, water moves in the small cells. Expansions are activated and cell elongate, stem elongate. So this is pretty much related to what we discussed in ordination and control. They have added this here. It's an integrated question. So let's move on. They have provided it with two varieties here, are two species of wild rice. They have given us the information, and from the information that is provided to us, we are told the two species fly at different times of the year. One of them is growing in water, but the other is capable of growing in dryness as well. These two rice species may have evolved by sympatric speciation, may have. So explain how both of them may have evolved through sympatric speciation. In sympatric speciation, we know there is no geographical barrier. So both the varieties among these, the first one, it grows in the wet environment, whereas the second one grows in the dry habitat, provided to us this inform. They have given this information in these bullets. So this allow ecological separation, since both of them will start at different years. So this is isolation, which is pre-zygotic. There is no gene flow occurring, and sympatric speciation occurred over times, and both these varieties were not able to interbreed. Now, let's take a question from Agent 2023, VA42. The sea blush is a flowering plant that grows on the west coast of North America. Individual sea blush plants produce fruit that is either winged or wingless. So this is winged and this is wingless. The dominant trait for fruit illegal and recessive is wingless. So we know this is dominant and this is recessive. You can see the size is 2 millimeter. This is scale bar is 2 millimeter. A large sample of sea blush was collected and the fruit wing characteristic was recorded. Name the type of variation. So the variation is disconnect variation because it's either winged or wingless. There are two discrete categories. There's no range of phenotype. We don't have something in between. So yeah, different sea blush plants can have different fruit colors. Different colors are possible. So there's two ways in which a new fruit color may naturally occur. So how naturally a different thing can occur? So we can recall color change is because of spontaneous mutation, because mutation can occur naturally, causing a different flower color to be produced. Codominant alleles can be there, they can get expressed as different color. We can't include epistasis here because the example is of a single gene. They have told to us that this basically is a single gene with two alleles, so epistasis cannot be included here. Early taxonomists classified this plant with winged end as different species to sea blush plant with wingless. Since this time, evidence of or from observation experiment has confirmed that the plants belong to the same species. So there's three examples of evidence obtained that help them to confirm that these plants belong to the same. How were they able to confirm that they actually are the same species? Because they both can breed together. Because whenever two organisms can breed together to produce fertile offspring, they are considered the same species. So they have the same number of genes, they have same alleles, different alleles might be there. Morphological features are common. Some amino acid and protein structures in both of these plants can be compared. Similarity in DNA can be compared using bioinformatics. Natural selection occurs in populations such as the population of seed plant. Explain why natural selection occurs in population. You need to give reasons why natural selection is required. So the facts of natural selection are we know that organisms overproduce. The organisms these show variation, they show different adaptations. Better variants get selected according to selection pressure. The allele frequencies are higher for favorable alleles. Better variants are more in number. They survive and reproduce more, and they pass favorable alleles to the next generation. So that's how we basically wrap up the concept of natural selection here. I hope this video was helpful for you. That's it from my side. Thank you so much.